metastasis. And the pressure is on me now. The longer I talk, the shorter the coffee break will be, so I will speed up the projection. Our aim was to follow how the cancer is going to spread and how it will be metastasis in animal models. The work was started 45 years ago in Szeged University. Szeged is in South Hungary, and I am now in Debrecen University. In Szeged, we have prepared 113 indium preparations, indium colloid, which accumulates, actually it is circulating with the blood, so we can visualize the heart. Indium colloid accumulated in the liver, that was recommended and used. We made several thousand scintigraphies, but is not used anymore because the 99 technetium took over. Indium iron macroaggregate caused microembolization. That's why it is counterindicated. Actually, the size would have been good for macroaggregation, but there were always present larger particles which can cause not only microemboli but emboli, and the patient could die. In the liver was perfect. I am not going to discuss it longer. This is a very old picture. This was the heart. It is a heart scintigraphy. The next is a normal liver scintigraphy. Probably you don't see too much of it. Then next one is a hepatomegaly. So if you can touch your liver, which means that you have a hepatomegaly. Here was a space-reducing process in the liver, which means that later it turned out that it was a cancer. We have also many small, so-called nanoparticles in the organism. For example, viruses, bacteria, yeast cells. I am going to talk about cancer cells and carbon particles, but there are many other particles. For example, we are also inhaling right now nanoparticles, dust and I don't know, smoke and so on. In Debrecen University, from the clinical isolates, it was a melanoma. We got a part of it, sliced it, digested with elastase, made a cell culture, and after 20 subculturing, we could name it that it was a cell line. So we made several cell lines, including hepatocarcinoma, nephroblastoma, myeloid leukemia, different myeloid leukemia, melanoma. D means Debrecen, so we call it, if you don't mind, head and Mida and so on. We used syngenic and xenograph models. Syngenic means that the suspension or the part tumor slice is taken from the same species, and of course it is implanted in the same species, or we can take clinical isolates and we can implant it in skid mice, and then of course we can also see the metastasis, how it develops. Local tumor formation was seen when the tumor was given locally, for example by subcutaneous implantation. Of course, that what we will see the tumor growth, which we can also